Hello and welcome to another episode of Momento Malum today. In this episode we'll be covering the genetic diversity of red raspberries throughout the world. Let's get started. The common red raspberry has been cultivated since the 4th century, and possibly even before then. The genetic improvement started in the development of local varieties, but by the 1800s, deliberate hybridization began, and many of the major varieties we see today were bred during this time period. Many of these varieties were selected from the European subspecies of the red raspberry or its American subspecies. This process occurred between the late 19th and early 20th century. The cultivars were derived from the early 1800s cultivars and their ancestors, as well as hybrids between both subspecies, and various chance seedlings found in the wild. Over time, several specific stud cultivars were selected to be used for parents multiple times. The result was that most of these cultivars are derived from a small number of parents. In particular, the cultivar Lloyd George. In the 1970s, Odivin calculated that 69% of all varieties released between 1930 and 1970 were at least partially derived from Lloyd George. With other cultivars such as Priashin, Newman, Lapham, Kubert, Newberg, Novus Kozuma, and several others were the major founders of the common red raspberry. The primordial cultivated red raspberries were self-incompatible, unable to self-pollinate. In recent years, mutations have been selected for that remove this self-incompatibility, allowing for self-pollination. Pedigree analysis has been used extensively as an estimate of the genetic diversity of the breeding populations and the level of inbreeding within said population. This combination of various algorithms has been used to study and estimate the gene pool of a specific population. To get a full understanding of the gene pool of the full domesticated red raspberry population, Dr. Adam Dale of the University of Guelph in the early 1990s would select 137 red raspberry varieties of known parentage and geographic origin. The gene pool was calculated using various algorithms and statistical methods such as the Ward's method to determine the relative different populations, the inbreeding level of different populations, and so on and so forth, with the assumption being that most of the open pollinated varieties were considered the result of self-pollination. The graph showcasing this data, in the form of tables, will be showcased in the last few slides. This will include a table made by the researcher, listing all the varieties they used, their country of origin, how frequently they are used in breeding, and an estimate of their genetic contribution. The next set of groups will cover the clusters of genetic groups within the framework of red raspberry cultivars, as well as the inbreeding coefficient of each individual cultivar. The larger the number, the more inbred it is. Hello, I just want to let you know, if you want to fund this channel, you can do so on the platforms seen in the description below. Of the 147 varieties of known parentage, all of them were derived from only 50 founding cultivars. The genetic coefficient ranged from less than 0.1% to 21%, depending on the cultivar, which means that some were used more often than others in the breeding process. Those with a greater genetic coefficient than 1% were 15 in number, 14 of which were introduced in the 19th and early 20th century, and all of which were derived from either the European or American subspecies. The 15th founding clone was called Creston, and is of unknown origin, although speculated to be 50% Lloyd George. It was found in the commercial field within British Columbia. 4 founding cultivars are in the pedigree of over 90 cultivars. These cultivar founders are Lloyd George, Hudson River Antwerp, English Globe, and Highland Hardy. The last 3 are found in only 2 cultivars that have been used as intermediate parents to crossbreed with other cultivars to make new varieties. These 2 are Cuthbert and Marlborough. 69% of all cultivars listed here are descended from Lloyd George, as 21% of the pedigree of 9% of the varieties are of Lloyd George origin. There's 21 founding clones amongst the 50 that have been used 7 or more times in the pedigrees, 3 of which are specimens of wild origin. This includes a specimen of Rubus orescens known as Oldenburg, one of the parents to make the Loganberry, an unnamed variety of the wild Rubus chamoris, and the black raspberry cultivar Cumberland. And in more recent years, 6 species have been introduced for specific purposes of breeding disease resistance or abiotic tolerance. A specimen of Rubus parafolius has been utilized for heat tolerance alongside a specimen of Rubus guinezensis. A specimen of Rubus cockenbarensis has been used in the breeding program to increase flower number, and Rubus odoratus has been utilized in the breeding of disease and pest resistance. It was found that through pedigree analysis, the varieties can be sorted into 13 genetic groups that are strongly related to a geographical origin and to a founder population, with the greatest separation being a small group of Russian varieties. All the Russian varieties tested were found in Group 1 and are derived from the cultivar Novost Kuzmina. The cluster 2 is found in the southern and central United States and is predominantly derived from equal parts Renair, Newman, and Lloyd George. Of these cultivars, all have at least minor traits of Renair, with the exception of one individual parent. The genetic contribution came from intermediate parents, such as Sunrise and September, in terms of the connection to Renair. And the contributions of Newman came through its descendants, Newberg, Taylor, Marcy, and Mullen Autumn Bliss. The most geographically diverse group is Cluster 3, and it's derived from Lloyd George, the experimental cultivar NY20990, and Herbert. This consists of 2 subgroups, the former consisting of Lloyd George, NY20990, and Herbert, of which are fall-bearing varieties, and the other group is derived from heritage, and all American fall-bearing raspberries have some degree of heritage, with the exception of Joe Mello. Joe Mello and heritage trace their lineage back to the cultivar Durham, and heritage/August Red is derived in part from NY20990 and Rosa chamoises. Joe Mello remains an unknown origin cultivar. In terms of summer-bearing cultivars, with the exception of Prestige, all are derived from Newberg, a hybrid between Newman and Herbert. Cluster 4 consists of mostly Northern European stock, with 2 American varieties mixed in, and these are mostly derived from Lloyd George, Amprosen, and Newberg. Group 5 consists of 2 cultivars derived from the founder cultivar Superlative. Group 6 consists of all varieties found in Central Europe. This group is mostly derived from Prussian, but 2 are derived mostly from Superlative. Group 7 consists of all British varieties, which can be traced back 13 generations, all of which have 7 founding cultivars within their pedigree: Lloyd George, Burnfolm, Pines Royal, Superlative, Hudson's River Antwerp, English Globe, and Highland Hardy. The exceptions to this rule be Mullen Autumn Bliss and Mullen Admiral. Cluster Group 8 is from the Pacific Northwest and contains 6 varieties that are crosses between the cultivar Cruston and various British selections. This cluster also contains 2 Danish varieties that have Pines Royal as at least one parent. Cluster Group 10 is derived from Lapham, consisting of 5 varieties of Lapham self-pollinated seedlings and other open-pollinated lines. Cluster Group 11 consists of varieties from North America in the most northern region, as well as parts of the USSR, France, and Hungary. It mostly consists of Lloyd George derivatives, and Hudson River Antwerp also makes up a decent chunk of the pedigree. The latter seems to be derived from intermediate parents such as Cuthbert and Viking. Cluster Group 12 contains Russian varieties that are derived from Usanka and Marlborough, and Cluster Group 13 consists of varieties almost wholly derived from rare or unique cultivars, with no pedigree in any of the major founder populations. Based on this information, it's suggested that the European and British populations are far different than most of the North American populations, and the Russian populations are vastly different than both. This indicates that crossbreeding between these three general geographic locations could increase the total genetic diversity by a considerable margin, as well as through the use of genetic diversity selected from the wild specimens, especially the American subspecies, which seems to be a lot more disease resistant overall. And that about covers everything. Thank you for watching.